Hello, my dear students. Good evening. Very good evening, everyone. Welcome to Sankalp Learning Classes. So, now last class ಅಲ್ಲಿ reproductive health chapter ಅನ್ನ complete ಮಾಡಿದ್ವಿ ಅಲ್ವಾ? So, we have studied about the reproductive health chapter. So, from today onwards, we are going to start the chapter Principles of Inheritance and Variation. That is the fourth chapter in your biology. Okay. Principles of inheritance and variation. I hope all are ready to listen the class. Yes, good evening, students. First, we should know the meaning of what is inheritance and what is variation, right? What is the meaning of inheritance and what is the meaning of variation, alva? So, namge inheritance andre enu, variation andre enu. So, we should know the meaning of that. Especially, so these two terms, so that belongs to the genetics. Here, what is genetics? So genetics, which deals with the inheritance as well as variation of characters from parents to the offspring. So first nodi, inheritance andre enu hagadre? You know that some characters, so that is transfer from parents to the next generation ಅಲ್ವಾ? ಸೊ ಪೇರೆಂಟ್ಸ್ ಇಂದ ನೀವು ಯಾವುದೇ ಆರ್ಗ್ಯಾನಿಸಮ್ ಅಲ್ಲಿ ನೋಡಿರ್ಬಹುದು ಅಥವಾ ಮನುಷ್ಯರಲ್ಲಿ ಇರಬಹುದು, ಬೇರೆ ಪ್ರಾಣಿಗಳಲ್ಲಿರ್ಬಹುದು, ಏನಾಗತ್ತೆ ಅಂದ್ರೆ ಕೆಲವೊಂದು ಕ್ಯಾರೆಕ್ಟರ್ಸ್, ಸೊ ದಟ್ ಈಸ್ ಪಾಸ್ ಫ್ರಮ್ ಇಟ್ಸ್ ಪೇರೆಂಟ್ಸ್, ರೈಟ್? ಸೊ ದಟ್ ಈಸ್ transmit from parents to the next generation. That is called as a inheritance. Inheritance is the process by which characters are passed on from parent to the progeny. Progeny means to the next generation. Okay. That is the meaning of the inheritance. Then what is variation? Variation means changes. It is the degree by which progeny differ from their parents. ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ, so some characters that is similar to your parents. So what you observe in you, so that is similar to your parents, but you are not exactly similar to your parents, right? So ನೀವು exact ಆಗಿ ನಿಮ್ಮ parents ತರನೇ ಇದ್ದೀರಾ? Are you exactly similar to your parents? No. So only few characters are transmitted from parents to the next generation, that is from parents to you, but there is a changes. You may observe, so you have some different characters or changes compared to your parents. That is called as the variation, alva? So is the degree by which progeny differ from their parents. So that is called as the variation. Here, about this inheritance and variation, the scientist who studied about the inheritance and variation, the scientist's name is Mendel, Gregor Johann Mendel. So the scientist who studied about the inheritance and he proposed the laws of inheritance. Okay, the scientist who studied about the inheritance and proposed the laws of inheritance, the scientist name is Mendel, that is Gregor Johann Mendel. The scientist who conducted the experiments on the garden peas for 7 years from 1856 to the 1863 and proposed the laws of inheritance. Here, for his experiments, Mendel selected the garden peas, pea plants. Why he selected the garden pea plants? So he can choose any other plants or any other organism, right? But why did he choose the garden pea plants? Because, so these pea plants, they have short lifespan. So one of the very important character, they have very short lifespan, means within 3 months they will grow and they bear the seeds and they die within 3 months. So only they are having the short lifespan, that is very helpful to study the different generation. So within a year, he can study more than 3 to 4 generations. Okay, so that is one. And they have some contrasting characters. So what are that contrasting characters that we will study later? The pea plants, they show the contrasting characters. And very easy to grow this plant. So there are many other characteristics. So that made the Mendel to choose the garden pea plant for his experiments. Then Mendel conducted artificial pollination or cross-pollination experiment using several true breeding pea plants. So he used the many pea plants for his pollination experiments. And what you mean by true breeding pea plants? What is the meaning of true breeding? Alba, true breeding andre enu? Here, true breeding means the pea plants, line means having undergone continuous cell pollination for several generation. So continuous agi cell pollination ಆಗ್ತಾ ಇರುತ್ತೆ. So continuously they will undergo the cell pollination. What is cell pollination? That you have studied in the first chapter, right? So these true breeding pea plants means continuously they undergo the cell pollination, not cross pollination. Okay, so that is called as the true breeding pea plant. Here, Mendel selected 14 true breeding pea plant varieties as pairs which are similar to except for one character with contrasting trait. Here, Mendel chose the different, observe the 7 different characters of the 7 different contrasting characters of the pea plant. Here, the first one is seed. So here, gray and round seed, white wrinkle seed, then yellow seed and the green seed, and flower. So white color flower and the violet color flower, two types, two contrasting type of the flowers are produced in pea plants. So that I told. And pod. So pod form, that is, it is full and it is constricted. It is like this, the pod shape, that is the constricted. And color of the pod, yellow and green. And stem, so axial pods and flowers along. Terminal, here the pods at the terminal and flowers at the top. And size of the plant, that is tall plant and the short plant. Okay, so these 7 contrasting characters he selected for his experiment. He observed contrasting characters, I told, right? So contrasting character anna pea plant alli observe ಮಾಡ್ತಾರೆ. Contrasting characters ಅಂದ್ರೆ ಏನು? Pea plant alli they may produce the yellow color seeds and some pea plants produce the green color seeds. That is the contrasting character. So ಈ ಎರಡು character ಅನ್ನ observe ಮಾಡ್ತಾರೆ. Flower ವೈಟ್ ಕಲರ್ ಫ್ಲವರ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಬಹುದು ಆರ್ ವೈಲೆಟ್ ಕಲರ್ ಫ್ಲವರ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುವಂತ ಒಂದು ಪ್ಲಾಂಟ್ಸ್ ಅನ್ನ he observed. So that is the contrasting character. So these 7 contrasting characters he selected for his experiments. Okay, then so very important for your exam, Mendel studied about the inheritance of one gene. Okay, so for this experiment, Mendel crossed tall and dwarf pea plants to study the inheritance of one gene. Very important for your exam. To study the inheritance of one gene, he crossed the tall pea plant, tall plant. So just imagine, this is the tall pea plant and dwarf, tall and dwarf pea plant for his experiment. Here, he collected the He collected the seeds produced as a result of this cross and grow them and generate the plants of the first hybrid generation, and this is called as the filial one progeny or F1 generation. When Mendel crossed the tall pea plant and the dwarf pea plant, so crossed means they are cross pollinated and produce the seeds, and that seeds are grown. ಆ seeds ಅನ್ನ ಮತ್ತೆ grow ಮಾಡಿದಾಗ ಏನು ಒಂದು plants produce ಆಯ್ತಲ್ಲ, so that is called as the F1 generation. Okay, F1 plants. And Mendel observed that all the F1 progeny plants are tall like of its parents, none were dwarf. What he observed in this, so all the plants which are grown in the F1 generation all are tall, but he crossed tall and dwarf plant, but none were the dwarf plant. Okay, he made similar observation for the other pairs of traits also, but he got the same result. Mendel then self-pollinated the tall F1 plants, and his surprise found that F2 generation, some of the offsprings were dwarf, and the character that was not seen in the F1 generation, so was now expressed. So in the F2 generation, so 1/4 of the plants of F2 generation are dwarf and 3/4 of the F2 generation plants were tall. So here, so let us study with here. So let us take the tall plant. Tall plants crossed with the dwarf plant. Okay, cross pollination. So here the plant which are produced, so here cross pollination occurred and the seeds are produced and the seeds are grown. So these are called as F1 generation, and in F1 generation all the plants what he grown, these all plants are tall plants. In the F1 generation all plants were tall. Okay, then again these tall plants are self pollinated. These F1 plants are self-pollinated. Self-pollination aitu. And these F1 plants, so here tall plant pollinated with the tall plant only. Okay, so self-pollinated. And in the F2 generation, what he observed? He observed tall plants as well as dwarf plants. Okay, in the F2 generation, what he observed? Tall plants, so 3/4 of the plants were tall and 1/4 of the plants were dwarf. So what is the reason behind this? Okay, so F2 generation nalli enaitu, all plants were, sorry, 1/4 of the plants were dwarf. So here, so only one of the parental traits was expressed in the F1 generation, while at the F2 stage, both traits were expressed in the proportion of 3:1. The contrasting traits did not show blending at either F1 or F2 stage. Here, there is no blending of the character. So tall ಮತ್ತೆ dwarf plant ಅನ್ನ cross ಮಾಡಿದಾಗ ಏನಾಗ್ಲಿಲ್ಲ ಅಲ್ಲಿ, so there is no blending, means intermediate form ಎಲ್ಲೂ form ಆಗ್ಲಿಲ್ಲ. Okay, so in the F1 and as well as F2 generation. Here, so Mendel's assumed that certain factors or genes, what we called now it as a gene, before it was called as a factor. Gene that transmit from parents to the next generation. The gene is transferred from parents to the next generation. Here, so if we take tall plant, if we represent tall plant as T, okay, so for each gene there are two, for each character two alleles will be there. So here tall plant, true breeding tall plant. True breeding tall plant means that is homozygous condition. Here true breeding tall plants means they are in the homozygous condition. ನಾನು ಇದನ್ನ TT ಅಂತ tall plant ಅನ್ನ TT ಅಂತ ತೆಗೆದುಕೊಳ್ತೇನೆ. So these are homozygous condition. Homozygous condition means, so this is the homozygous condition. And dwarf plant, that is also true breeding dwarf plant. So that is, so I will not take D, I will take small t, small t. It is also in homozygous condition. Okay. Homozygous condition means which is having the same alleles. Okay. Then ಈಗ ನೋಡಿ ನೆಕ್ಸ್ಟ್ ಏನಾಗತ್ತೆ ಅಂದ್ರೆ F1 ಜನರೇಷನ್, ಇಲ್ಲಿ ಗ್ಯಾಮೇಟ್ಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗತ್ತೆ ಅಲ್ವಾ, ಗ್ಯಾಮೇಟ್ಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗತ್ತೆ. Gametes production, each gamete that is having tall plant gamete that is having T or capital T, and dwarf plant gametes that is having the small t or small t because of meiosis alva. Meiosis agodake enagatte diploid irodu haploid agatte alva. And in the F1 generation what happens? Capital T, so when it combines with the capital T, small t, and capital T, small t, then this one capital T, small t, and this one with one capital T, small t. Okay. So here what he observed, in the F1 generation all plants were tall plants. There is no dwarf plants. But what you observe here, capital T, small t, capital T, small t, right? So ಇಲ್ಲಿ capital T ಮತ್ತೆ small t ಕಾಣಿಸ್ತಾ ಇದೆ ಅಲ್ವಾ? But these are not a dwarf plants because here capital T is dominant over the small t. So capital T is expressed, so it is the tall plant alva. So ಈಗ ಇದನ್ನ heterozygous condition ಅಂತ ಕರಿತೇವೆ ನಾವು. This is called as heterozygous condition. So parental plants were, they are tall. So here in the F1 generation all the plants are similar to parental generation, but their genotype, that is capital T, small t. Okay, but here capital T is expressed, small t is suppressed. Here capital T is called as dominant over the small t. Okay, so this is about the F1 generation. Next nodi F2 generation alli enagatte anta, in the to obtain the F2 generation, F1 plants are self-pollinated. Here capital T crossed with capital T, small t. Tall plant, both are tall plants, they are crossed. So what you get in the F2 plants? Here the gametes produced capital T, small t, and here also capital T, small t gametes. Okay, so here when this capital T combines with the capital T, so capital T, then this capital T with the small t, capital T, small t, and here also capital T, small t, and last one small t, small t. So the 4 plants were obtained. Here 3/4 means 3 plants were tall and 1 plant that is dwarf alva. 1 plant enagittu, dwarf agittu, that is small t, small t. So this is the inheritance of one gene according to the Mendel. Nodi, here this is the tall plant which is represented as genotype is capital T, and this is the small t, dwarf plant. So gametes produced, that is capital T, small t. So F1 plants, capital T, small t, what is this called? This is called as heterozygous condition, but capital T is expressed, small t is not expressed. So it is capital T is dominant over the small t. Okay. Here, when the F2 F1 plants are crossed, nodi. So this is having the capital T small t, and this plant is also having the capital T small t. When these plants are crossed, here total 4 plants were obtained. In that 3 plants are tall plants and 1 plant is dwarf plant. But what is the genotypic ratio? Here genotypic means capital T, 1 plant is there, and capital T, small t, 2 plants, and small t, 1 plant. Okay, 1 is to 2 is to 1. Genotypic ratio. Phenotype. Genotype means the genetic makeup of these plants, and phenotype means external characters. Genotype, phenotype andre nenpitkolli. Phenotype, that is the external characters. So phenotypic ratio is 3 is to 1. Very, very important for your exam. Okay, I hope you got it. So, Mendel used the Punnett square to express inheritance of one gene. So, Punnett square anna use ಮಾಡಿ. So, easily we can get the F2 generation plants. Okay. So, again, parental generation, tall, crossed with dwarf. Okay. So genotype is capital T, and here small t, small t. The gametes produced capital T here and small t here. F1 generation plants. So here capital T, small t. All are heterozygous tall plants. Okay. To obtain the F2 generation, we can use the Punnett square. So what is this checkerboard or Punnett square? Capital T crossed with capital T, capital T. Okay. So let us choose the Punnett square. Nodi, ಈ ತರ ಚಕ್ಕರ್ ಬೋರ್ಡ್ ಅನ್ನ ಯೂಸ್ ಮಾಡಿ ಇಲ್ಲೇನ್ ಮಾಡಬೇಕು, capital T gametes ಅನ್ನ ಬರಿಬೇಕು, small t, and here capital T, capital T, here small t. Okay, so just multiply this T, capital T, and idanna add ಮಾಡಿ ಬರೀರಿ, capital T, small t, then small t, capital T, so that is capital T, small t, last one small t, small t. Okay, so here also you can get the capital T, capital T, capital T, small t, capital T, small t, and small t, small t. Easily you will get the answer. Okay, so this is the answer. So ಈ ರೀತಿಯಾಗಿ Punnett square ಅನ್ನ ಯೂಸ್ ಮಾಡಿಕೊಂಡು inheritance of one gene. So very important for your exam. Okay. Next, test cross. What is test cross? Here, when F1 hybrid is crossed back with the recessive parent, it is known as a test cross. It is used to know the genotype of the given plant or the animal. So here, in the F2 generation, ಈಗ ಏನಾಗಿದೆ, F1 plants ನಿಮಗೆ ಗೊತ್ತಿದೆ, that is the true breeding. capital T ಇದೆ, small t, small dwarf plant, small t ಇದೆ ಅಂತ ನಿಮಗೆ ಗೊತ್ತಿದೆ ಅಲ್ವಾ. ಬಟ್ F2 generation ಅಲ್ಲಿ capital T, small t ಇದೆಯಾ ಅಥವಾ small t capital T ಇದೆಯಾ ಅನ್ನೋದನ್ನ. So is it homozygous or the heterozygous? So that we can find out by doing the test cross. So idanna find out ಮಾಡಬಹುದು. F2 generation plants ಅಲ್ಲಿ are they homozygous or heterozygous? Homozygous condition ಅಲ್ಲಿ ಇದೆಯಾ, heterozygous condition ಅಲ್ಲಿ ಇದೆಯಾ ಅಂತ find out ಮಾಡಬಹುದು. Okay, yes, here for example, capital T, tall plant, capital T, crossed with the small t. Okay. So we can find out the F1 hybrid is crossed back with the recessive parent. It is known as the test cross. So here F1 plants. So you don't know F1 plants are heterozygous or homozygous. Alva. So just take the heterozygous condition, capital T, small t crossed with small t. Okay. So small t is the recessive parent. Ignore capital T, small t and capital T, small t, small t, small t, and small t, small t. So here 50% tall plants and 50% dwarf plants are obtained. Then you can say F1 plants are heterozygous condition. 50% ಏನಾಗಿದೆ tall plant ಇದೆ, 50% dwarf plant ಇದೆ. Then you can say it is the ಹಿಟಿರೋಝೈಗಸ್ ಕಂಡೀಷನ್. ಅಕಸ್ಮಾತ್ ಆಗಿ ಹೋಮೋಝೈಗಸ್ ಕಂಡೀಷನ್ ಇದ್ರೆ ಏನಾಗ್ತಿತ್ತು, ಕ್ಯಾಪಿಟಲ್ ಟಿ ಕ್ರಾಸ್ ವಿತ್ ಸ್ಮಾಲ್ ಟಿ ಆದ್ರೆ ಏನಾಗ್ತಿತ್ತು, all plants will be tall ಅಲ್ವಾ, all plants were tall, but here F1 plants are heterozygous condition. We can find out by using the test cross ಅಂದ್ರೆ ಏನು ಇಲ್ಲ, F1 hybrid. Okay, F1 plants crossed with recessive parents, so that is known as a test cross. By using these experiments, Mendel first law, that is the law of dominance, was proposed. So what it states, law of dominance, what it states, okay, law of dominance ಏನನ್ನ ಹೇಳುತ್ತೆ, characters are controlled by discrete units called factors. So the characters, they are controlled by some units that is called as factors, and factors occur in pairs. In a dissimilar pair of factors, one member of the pair is dominates the other. So other is the recessive. Here factors occur in pairs. Okay. So for example, capital T or capital T, small t. So these are called as factor alleles in dissimilar pair. So this is the dissimilar pair. So one member of the pair dominates the other recessive. Okay, so that is the law of dominance. Then law of segregation. What it states, law of segregation. ನೋಡಿ, ಮೆಂಡಲ್ಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಬಗ್ಗೆ ನೀವು SSLC ಅಲ್ಲಿ ಕೂಡ ಸ್ಟಡಿ ಮಾಡಿರ್ತೀರಾ ಅಲ್ವಾ. ಸೊ ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಮೆಂಡಲ್ಸ್ 1 ಜೀನ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಆಯ್ತು ಅಲ್ವಾ, ಇಲ್ಲಿ ಲಾ ಆಫ್ ಡಾಮಿನೆನ್ಸ್ ಅನ್ನ ಪ್ರಪೋಸ್ ಮಾಡಿದ್ರು. ಅಂಡ್ ಲಾ ಆಫ್ ಸೆಗ್ರಿಗೇಷನ್, ವಾಟ್ ಈಸ್ ಲಾ ಆಫ್ ಸೆಗ್ರಿಗೇಷನ್? The alleles do not show the blending and that both the characters are recovered as such as F2 generation through one of this is not seen in the F1 stage. The parents contain two alleles during the gamete formation. The factors or alleles of a pair segregate or separate from each other such that gametes receives only one of the two factors. So here parents contain two alleles during the gamete formation. Okay. So here during the gamete formation, so for yellow seed ಗೆ ಕ್ಯಾಪಿಟಲ್ ವೈ ಅಂತ ಇದೆ, ಹಾಗೆ ಗ್ರೀನ್ ಸೀಡಿಗೆ ಸ್ಮಾಲ್ ವೈ ಅಂತ ಇದೆ. ಓಕೆ, ಸೊ ದೀಸ್ ಆರ್ ದಿ ಟೂ ಅಲೀಲ್ಸ್. ಸೊ ಡ್ಯೂರಿಂಗ್ ದ ಗ್ಯಾಮೆಟ್ ಫಾರ್ಮೇಶನ್ ದೇ ಸೆಗ್ರಿಗೇಟ್. ಸೊ ಇಲ್ಲಿ ನೋಡಿ, ಗ್ಯಾಮೆಟ್ ಫಾರ್ಮೇಶನ್ ಆಗ್ಬೇಕಾದ್ರೆ ಏನಾಗತ್ತೆ, ದೇ ಸೆಪರೇಟ್, ರೈಟ್? ಸೊ ಹಿಯರ್ ಕ್ಯಾಪಿಟಲ್ ವೈ, ಸ್ಮಾಲ್ ವೈ ಇಸ್ ದರ್, ಮೀನ್ಸ್ ದೇ ಸೆಪರೇಟ್ ಡ್ಯೂರಿಂಗ್ ದ gamete formation. Here you can see the segregation of the gametes from each other such as gamete receives only one of the two factors. The gametes, it may receive capital Y or small y. Alva. Homozygous parent produces all gametes that are similar. It contains same type of the allele. Heterozygous parents produces two kinds of gametes, each having one allele with the equal proportion. ಇಲ್ಲಿ ನೋಡಿ, capital Y. So I will take tall and short only. So tall plants capital T ಇದೆ, small t ಇದೆ. So these are homozygous condition. Homozygous condition ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? Always gametes contain capital T, here small t. Okay. But in the heterozygous condition, ಈಗ capital T small t ಇದೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. Gametes either it may contain capital T or small t, only one. Okay, not both. ಎರಡು alleles ಅನ್ನ, so these two factors will be not present in the gamete. During the gamete formation, they segregate, they separate. So that is the law of segregation. ಅಲ್ವಾ? ನೀವು F1 ಒನ್ ಜೀನ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡಿದ್ರಿ ಅಲ್ವಾ, ಯಾವ ರೀತಿಯಾಗಿ ಗ್ಯಾಮೆಟ್ ಸೆಪರೇಟ್ ಗ್ಯಾಮೆಟ್ ಫಾರ್ಮೇಶನ್ ಆಗಬೇಕಿದ್ರೆ, ಸೊ ದೀಸ್ ಫ್ಯಾಕ್ಟರ್ಸ್ ಆರ್ ಅಲಿಲ್ಸ್ ದೇ ಸೆಗ್ರಿಗೇಟ್ ಆರ್ ಸೆಪರೇಟ್ ಫ್ರಮ್ ಈಚ್ ಅದರ್. ಈಗ ಕ್ಯಾಪಿಟಲ್ ವೈ ಇದ್ರೆ ಅಥವಾ ಕ್ಯಾಪಿಟಲ್ ವೈ ಸ್ಮಾಲ್ ವೈ ಇದ್ರೆ ಒನ್ ಗ್ಯಾಮೆಟ್ ದಟ್ ಕಂಟೈನ್ ಕ್ಯಾಪಿಟಲ್ ವೈ ಅಂಡ್ ಅನದರ್ ಗ್ಯಾಮೆಟ್ ದಟ್ ಕಂಟೈನ್ ದ ಸ್ಮಾಲ್ ವೈ. ಸೊ ದಟ್ ಈಸ್ ದ ಲಾ ಆಫ್ segregation. So here, according to the Mendel's law of dominance. So law of dominance, there is no blending of the characters. So there is no intermediate characters will be formed. Either it may be dominant or it may be recessive. So like that it is alva, ಒಂದು dominant ಇರುತ್ತೆ ಅಥವಾ recessive ಆಗಿರುತ್ತೆ. So but here exceptions of conclusions of Mendel. There are two exceptions of law of dominance. So two exceptions means which do not obey the law of dominance. Mendel en ಮಾಡ್ತಾರೆ, law of dominance ಅನ್ನ propose ಮಾಡ್ತಾರೆ ಅಲ್ವಾ. But here these two incidents, these two things, so they will not obey the law of dominance. The first one is incomplete dominance and the codominance. Let us study about the incomplete dominance and the codominance. Okay. According to Mendel's law of dominance, dominant character must be present in F1 generation, but in some organism, F1 generation is different from both of the parents, and both factors such as dominant and recessive are present in the incomplete dominance. But dominant factors are unable to express their characters completely, resulting in the different type of generation which is different from both of the parents. ಸೊ ಇಲ್ಲಿ ಮೆಂಡಲ್ಸ್ ಲಾದಲ್ಲಿ ಏನಾಗಿತ್ತು, ಸೊ ದೇ ಆರ್ ದ ಪ್ರೊಜೆನೀಸ್, ದ plants which are produced in the F2 generation, they are similar to their parents. Parents ಗೆ ಸಿಮಿಲರ್ ಆಗಿತ್ತು ಅಲ್ವಾ, ಒಂದು ಟಾಲ್ ಇತ್ತು ಅಥವಾ ಡಾರ್ಫ್ ಇತ್ತು. ಬಟ್ ಹಿಯರ್ some exceptions are there, they will not obey the law of dominance. Here intermediate species are formed, intermediate characters are formed which are not similar to parental generation. Okay, so here where we can observe the incomplete dominance, so here Mirabilis jalapa, that is 4 o'clock plant, and snapdragon plant. Okay, snapdragon or the dog plant, it is also called as dog plant. So here we can observe the incomplete dominance. So let us study in detail about the incomplete dominance. Here 3 different types of plants are found in Mirabilis on the basis of flower color such as red and white and pink. Okay, when the plants with red flowers crossed with the white flowers plants, pink flower is obtained in the F1 generation. The reason of this is that genes red color is incompletely dominant over the genes of the white color. So here incomplete dominance. Okay, so Mirabilis jalapa, 4 o'clock plant, or snapdragon or the dog plant. Okay, so here it produces the 2 types of flower, so 3 types of flower. Here kept red flower, so red flower that is having the dominant capital R, and white flower that is having the alleles, that is small r. Okay. So here F1 generation plants, they are having the capital R, small r. So according to Mendel's law, so F1 generation plants ಏನಾಗಬೇಕಿತ್ತು, they should produce the red color flower ಅಲ್ವಾ? ಹೌದು ಅಲ್ವಾ? ಓಕೆ, ಇಲ್ಲಿ ಏನಾಗತ್ತೆ ಅಂದ್ರೆ F1 ಪ್ಲಾಂಟ್ಸ್ ಅಂದ್ರೆ ಫಸ್ಟ್ ಫೀಲಿಯಲ್ ಜನರೇಷನ್ ಪ್ಲಾಂಟ್ಸ್. ಓಕೆ, F1 ಜನರೇಷನ್ ಇಲ್ಲಿ ಏನಾಗ್ಬೇಕಿತ್ತು, ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಮತ್ತೆ ಸ್ಮಾಲ್ ಆರ್ ಇದೆ. ಅಂದ್ರೆ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಇದೆ ಅಂದ್ರೆ ಏನಾಗ್ಬೇಕಿತ್ತು, ರೆಡ್ ಫ್ಲವರ್ ಆಗ್ಬೇಕಿತ್ತು ಅಲ್ವಾ? ಈಗ ನಾವು ಟಾಲ್ ಮತ್ತೆ ಡ್ವಾರ್ಫ್ ಅನ್ನ ಸ್ಟಡಿ ಮಾಡಿದಾಗ ಕ್ಯಾಪಿಟಲ್ ಟಿ ಸ್ಮಾಲ್ ಟಿ ಇದ್ದಾಗ ಏನಾಗಿತ್ತು, ದ ಪ್ಲಾಂಟ್ಸ್ ಆರ್ ಟಾಲ್, ಟಾಲ್ ಪ್ಲಾಂಟ್ಸ್ ಆಗಿತ್ತು ಅಲ್ವಾ? ಸೊ ಇಫ್ ಯು ಆರ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್ ಆರ್ ನಾಟ್, ಪ್ಲೀಸ್ ಕಮೆಂಟ್ ಇನ್ ದ ಕಮೆಂಟ್ ಬಾಕ್ಸ್. ಏನಾದ್ರೂ ಡೌಟ್ ಇದ್ರೆ ನೀವು ಇಲ್ಲಿ ಕೇಳ್ಬಹುದು. ಓಕೆ, ಇಲ್ಲಿ ನೋಡಿ ಏನಾಯ್ತು, F1 ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಏನಾಯ್ತು, ರೆಡ್ ಫ್ಲವರ್ ಫಾರ್ಮೇಶನ್ ಆಗ್ಬೇಕಿತ್ತು. ಬಟ್ ಹಿಯರ್ this capital R is not completely dominant over the small r. ಇಲ್ಲಿ capital R ಏನಾಗಿರೋದಿಲ್ಲ, ಕಂಪ್ಲೀಟ್ ಆಗಿ ಡಾಮಿನೆಂಟ್ ಆಗಿಲ್ಲ. capital R ಡಾಮಿನೆಂಟ್ ಆಗಿಲ್ಲ ಸ್ಮಾಲ್ ಆರ್ ಮೇಲೆ. ಸೊ ಇಂಟರ್ಮೀಡಿಯೇಟ್, ರೆಡ್ ಅಲ್ಲ ವೈಟ್ ಅಲ್ಲ, ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಕಲರ್ ಈಸ್ ಫಾರ್ಮ್, ದಟ್ ಇಸ್ ದ ಪಿಂಕ್ ಕಲರ್ ಫ್ಲವರ್. ಈಗ ನೋಡಿ F2 ಜನರೇಷನ್ ಪ್ಲಾಂಟ್ ಅನ್ನ, ಸೊ F2 ಜನರೇಷನ್ ಪ್ಲಾಂಟ್ಸ್ ಅನ್ನ ಹೇಗೆ ಆಬ್ಟೈನ್ ಮಾಡೋದು, ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಪಿಂಕ್ ಕಲರ್ ಫ್ಲವರ್ ಪ್ರೊಡ್ಯೂಸಿಂಗ್ ಪ್ಲಾಂಟ್ ಕ್ರಾಸ್ಡ್ ವಿತ್ ದ ಸೇಮ್ ಸೆಲ್ಫ್ ಪಾಲಿನೇಟೆಡ್. ಇಲ್ಲಿ ಏನಾಗತ್ತೆ, ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಸ್ಮಾಲ್ ಆರ್, ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಸ್ಮಾಲ್ ಆರ್, ಗ್ಯಾಮೆಟ್ಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಯ್ತು ಅಲ್ವಾ? ಗ್ಯಾಮೆಟ್ಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಯ್ತು. ಹಿಯರ್ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಕ್ಯಾಪಿಟಲ್ ಆರ್, capital R small r, capital R small r, and small r small r. So can you say how many red color plants, flower producing plants, how many white and how many pink color flower producing plants are obtained in the F2 generation? ಸೊ ಹೇಳ್ಬೋದಾ, F2 ಜನರೇಷನ್ ಅಲ್ಲಿ ಎಷ್ಟು ರೆಡ್ ಕಲರ್ ಫ್ಲವರ್ ಫಾರ್ಮೇಶನ್ ಮಾಡುವಂತ ಪ್ಲಾಂಟ್ಸು, ದೆನ್ ಪಿಂಕ್ ಕಲರ್ ಅಂಡ್ ವೈಟ್ ಕಲರ್ ಫಾರ್ಮೇಶನ್ ಮಾಡುವಂತ ಪ್ಲಾಂಟ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿದೆ ಅಂತ ಹೇಳ್ಬೋದಾ, ಇನ್ ಇನ್ಕಂಪ್ಲೀಟ್ ಡಾಮಿನೆನ್ಸ್? ಪ್ಲೀಸ್ ಕಮೆಂಟ್ ಇನ್ ದ ಕಮೆಂಟ್ ಬಾಕ್ಸ್. ಓಕೆ, ಇಲ್ಲೇನಾಗಿದೆ, ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಇದ್ರೆ ಮಾತ್ರ ಏನು, ದಟ್ ಈಸ್ ದ ರೆಡ್ ಫ್ಲವರ್. ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಸ್ಮಾಲ್ ಆರ್ ಇದ್ರೆ ದಟ್ ಈಸ್ ದ ಪಿಂಕ್, ಅಂಡ್ ಸ್ಮಾಲ್ ಆರ್ ಇದ್ರೆ ದಟ್ ಈಸ್ ದ ವೈಟ್ ಕಲರ್ ಫ್ಲವರ್. ಸೊ ಹಿಯರ್ ರೆಡ್ ಕಲರ್ ಫ್ಲವರ್ that is not able to dominate over the white color. So it is called as the incomplete dominance. So this is the exceptions of Mendel's law of dominance. Okay, very important. Second one is the codominance. So this is also exception for Mendel's law of segregation. Okay, Sujata, very good. Red color flower 1 and pink color flower 2. Yes. And how many white color flowers? Yes, very good. Sujata, very good. Codominance. In codominance, neither allele is completely dominant and both alleles are expressed in the phenotype. Illi, both alleles are expressed. ಸೊ ಮೆಂಡಲ್ಸ್ ಲಾದಲ್ಲಿ ಏನಾಗಿತ್ತು, ಓನ್ಲಿ ಒನ್ ಡಾಮಿನೆಂಟ್ ಇರೋದು ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗ್ಬೇಕಿತ್ತು ಅಲ್ವಾ? ಬಟ್ ಹಿಯರ್ ಬೋತ್ ಆರ್ ಎಕ್ಸ್ಪ್ರೆಸ್ಡ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ABO ಬ್ಲಡ್ ಗ್ರೂಪಿಂಗ್ ಇನ್ ಹ್ಯೂಮನ್ಸ್. ಸೊ ಲೆಟ್ ಅಸ್ ಟೇಕ್ ದ ಎಕ್ಸಾಂಪಲ್ ಆಫ್ ABO ಬ್ಲಡ್ ಗ್ರೂಪಿಂಗ್. ಹಿಯರ್ ಇನ್ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ಸ್ ಅಲ್ಲಿ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ ಬ್ಲಡ್ ಗ್ರೂಪ್ಸ್ ಇದೆ ಅಲ್ವಾ? A ಬ್ಲಡ್ ಗ್ರೂಪ್, B, and AB O blood group ಇದೆ ಅಲ್ವಾ? And here this blood group of human beings controlled by gene I. Okay, so this blood group is controlled by gene I. Here this I gene, it is having 3 alleles. So I gene that is having IA, IB and small I. Okay, so these are the alleles of this I gene. Okay, ಈ IA gene andre ಏನು? Here around the surface of the RBC ಮೇಲ್ಗಡೆ sugar polymers ಇರುತ್ತೆ. RBC on the plasma membrane of the RBC, red blood cells ಅಲ್ವಾ? ಈ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಮೇಲ್ಗಡೆ ಶುಗರ್ ಪಾಲಿಮರ್ಸ್ ಇರುತ್ತೆ. ದಟ್ ಈಸ್ ಡಿಫ್ರೆಂಟ್ ಇನ್ IA ಜೀನ್ ಇದ್ರೆ ಡಿಫ್ರೆಂಟ್ ಇರುತ್ತೆ, IB gene idre, so that is different. Okay, that sugar polymer is different. Okay, ಇಲ್ಲಿ ನೋಡಿ, now if the allele form, okay, so if the parent is having the allele IA allele ಇದೆ, ಹಾಗೆ IA, okay, IA allele is there. So ಇಲ್ಲಿ ಗ್ಯಾಮೆಟ್ ಫಾರ್ಮೇಶನ್ ಆಗ್ಬೇಕಿದ್ರೆ IA allele ಇದ್ರೆ, so here you can see IA idre, the blood group name is A. Okay, so IA is dominant. ಈಗ IA ಏನಾಯ್ತು? ಡಾಮಿನೆಂಟ್ ಆಯ್ತು. ಈಗ IB ಇದ್ರೆ ಯಾವುದು ಡಾಮಿನೆಂಟ್, ಯಾವುದು ರಿಸೆಸಿವ್? Here IA IB are both are dominant, but small I, that is the recessive one. Okay, ಈಗ ನೋಡಿ IA ಇದೆ, both are similar and the blood group is A. If IA IB is there, IA IB ಇದ್ರೆ ಯಾವ ಬ್ಲಡ್ ಗ್ರೂಪ್ ಇರುತ್ತೆ? So that is both are expressed. So it is called as AB blood group. AB blood group ಅಲ್ಲಿ ಏನಾಗಿದೆ ಅಂದ್ರೆ I gene that is having the IA gene also and IB also. IA IB ಎರಡು ಇದೆ, ಎರಡು ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಿದೆ. So that is called as the codominance. ಯಾವ್ದಾದ್ರು ಒಂದೇ, ಈಗ AB ಬ್ಲಡ್ ಗ್ರೂಪ್ ಆಗಿದೆ ಅಲ್ವಾ, ಐದರ್ A ಆರ್ B ಬ್ಲಡ್ ಗ್ರೂಪ್ ಆಗ್ಬೇಕಿತ್ತು, ಯಾವ್ದಾದ್ರು ಒಂದೇ ಡಾಮಿನೆಂಟ್ ಆದ್ರೆ, A ಡಾಮಿನೆಂಟ್ ಓವರ್ B ಆದ್ರೆ A ಬ್ಲಡ್ ಗ್ರೂಪ್ ಆಗ್ಬೇಕಿತ್ತು ಅಲ್ವಾ, ಆದ್ರೆ ಇಲ್ಲೇನಾಗ್ತಾ ಇದೆ, ಬೋತ್ ಆರ್ ಡಾಮಿನೆಂಟ್ ಹಿಯರ್. ಸೊ ದ ಬ್ಲಡ್ ಗ್ರೂಪ್ ಈಸ್ AB. ಸೊ ದ ಬ್ಲಡ್ ಗ್ರೂಪ್ ಈಸ್ AB. ಈಗ IA ಸ್ಮಾಲ್ I ಇದೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ, IA ಸ್ಮಾಲ್ I, then this capital A is dominant over this small I, so the blood group is A. Okay, so ಈಗ B ಇದ್ರೆ ಏನಾಗುತ್ತೆ, the blood group is IB, small I idre the blood group is B. If small I is present, there is no sugar polymers present around the RBC, so the blood group name is O blood group. Okay, so this is the codominance, multiple allelism. It is also called as multiple allelism. ಈಗ ನೋಡಿ ಇನ್ನೊಂದ್ಸಲ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೇನೆ. So just recall the Mendel's law. You know the incomplete dominance, where the red color flower is not able to dominate over the white color alva, incomplete dominance ಆಯ್ತು. codominance ಅಂದ್ರೆ both are expressed. Here both characters are expressed. For example, ABO blood grouping in human beings. Here I gene that is having the alleles IA, IB and small I ಇದೆ. Okay, if the person is having the IA gene, then the blood group is A. Okay, but the person is having the IA, IB, the blood group is AB. Here, both are expressed. ಇಲ್ಲಿ ಎರಡು ಕೂಡ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗಿದೆ ಅಲ್ವಾ? So it is called as the codominance. I hope ಅರ್ಥ ಆಗಿದೆ ಅಂತ ಅನ್ಕೊಳ್ತೇನೆ. ಅರ್ಥ ಆಗಿದ್ಯಾ? Please comment in the comment box. ಅರ್ಥ ಆಗಿದ್ಯಾ ಅಂತ ಕಮೆಂಟ್ ಮಾಡಿ. Then, if the IA small I is present, then the blood group is A because this small I is the recessive. Okay. So this is also example for the multiple allelism. So the example of ABO blood grouping that produces the good example for the multiple alleles means more than 2 alleles ಇದೆ ಅಲ್ವಾ? ಈ ಕಾಮನ್ ಆಗಿ ಟಾಲ್ ಗೆ ಏನಿರ್ತಿತ್ತು? ಕ್ಯಾಪಿಟಲ್ ಟಿ ಸ್ಮಾಲ್ ಟಿ ಎರಡೇ ಅಲಿಲ್ಸ್ ಇದೆ ಅಲ್ವಾ? ಬಟ್ ಇಲ್ಲಿ ಬ್ಲಡ್ ಗ್ರೂಪ್ ಗೆ ಏನಾಗಿದೆ? ಸ್ಮಾಲ್ ಕ್ಯಾಪಿಟಲ್ ಐ ಎ, ಐ ಬಿ ಅಂಡ್ ಸ್ಮಾಲ್ ಐ ಇದೆ ಅಲ್ವಾ? ಮೋರ್ ದನ್ 3 alleles are governing the same character, so it is also called as the multiple allelism. Here, a single gene product may produce more than one effect. So that is called as the multiple allelism. Here, starch synthesis in pea seeds is controlled by one gene. It has 2 alleles, B and capital B and small b. Starch is synthesized effectively by capital B homozygote and therefore large starch grains are produced. So, ಇಲ್ಲಿ pea plant ಅಲ್ಲಿ capital B ಇದ್ರೆ large size grains are produced because of the starch synthesis. Okay. Then if BB homozygous has less efficiency, hence produce the smaller grains. Small b ಇದ್ರೆ small grain ಅನ್ನ produce ಮಾಡಿತ್ತು. After maturation of seeds, BB seeds are round and the BB seeds, small b seeds are wrinkled. Heterozygous produces the round seeds and so B seems to be a dominant allele, but the starch grains produced are the intermediate size. Capital B, small b. Here, the starch is present is intermediate. Capital B ಏನಾಗಿತ್ತು? starch ಏನಾಗಿತ್ತು? ಜಾಸ್ತಿ ಇತ್ತು. Small b ಅಲ್ಲಿ ಕಡಿಮೆ ಇತ್ತು. But in case of capital B, small b, the starch is intermediate. If the starch grain size is considered as the phenotype, then from this angle, the alleles show the incomplete dominance. ಇಲ್ಲೂ ನಾವು starch synthesis ಮೇಲೆ ಸ್ಟಡಿ ಮಾಡೋದಾದ್ರೆ, so this capital B, small b, that is having the incomplete dominance. Okay. So that is about the exceptions of the Mendel's law. Then we have to study about the inheritance of 2 gene. ಈಗ ನಾವು ಏನ್ ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ? inheritance of 1 gene ಅಲ್ವಾ? 1 gene ಅಂದ್ರೆ only one character we have taken. Only height of the plant, tall plant ಮತ್ತೆ dwarf plant ಒಂದೇ ಕ್ಯಾರೆಕ್ಟರ್ ಅನ್ನ ತೆಗೆದುಕೊಂಡಿದ್ವಿ ಅಲ್ವಾ? either it is tall plant or dwarf plant. So ಈ ತರ ತೆಗೆದುಕೊಂಡಿದ್ವಿ. But here we have to take the 2 characters. So it is inheritance of 2 gene. It is also called as dihybrid cross. Okay. Inheritance of 2 gene, that is also called as dihybrid cross and law of independent assortment. So we can explain the law of independent assortment using this dihybrid cross. What is law of independent assortment? Idanna last ಗೆ ಸ್ಟಡಿ ಮಾಡೋಣ. Let us take the 2 characters combined in a to form the hybrid. So ಇಲ್ಲಿ ನಾವೇನ್ ತೆಗೆದುಕೊಂಡಿದ್ದೇವೆ? For dihybrid cross, yellow round seed producing pea plant, yellow capital Y round seed producing pea plant, yellow color round seed ಅನ್ನ produce ಮಾಡುವಂತ pea plant and green wrinkle seeds, ಗ್ರೀನ್ ಕಲರ್ ಸೀಡ್ಸ್ ಬಟ್ ರಿಂಕಲ್ ರೌಂಡ್ ಅಲ್ಲ, ರಿಂಕಲ್ ಆಗಿರುವಂತ ಸೀಡ್ಸ್, ಸೀಡ್ಸ್ ಪ್ರೊಡ್ಯೂಸಿಂಗ್ ಪ್ಲಾಂಟ್ ಅನ್ನ ಕ್ರಾಸ್ ಮಾಡಿದ್ದಾರೆ ನೋಡಿ. ಕ್ಯಾಪಿಟಲ್ ವೈ ಯೆಲ್ಲೋ ರೌಂಡ್, ಕ್ಯಾಪಿಟಲ್ ಆರ್ ರೌಂಡ್ ಸೀಡ್ಸ್ ಕ್ರಾಸ್ ಮಾಡಿದ್ದಾರೆ. ಯಾವ್ದ್ರ ಜೊತೆ? ಗ್ರೀನ್ ಕಲರ್. ಯೆಲ್ಲೋ ಕಲರ್ ಅಂದ್ರೆ ಕ್ಯಾಪಿಟಲ್ ವೈ ತೆಗೆದುಕೊಂಡಿದ್ದೇವೆ, ಗ್ರೀನ್ ಕಲರ್ ಅಂದ್ರೆ ಸ್ಮಾಲ್ ವೈ ಸ್ಮಾಲ್ ಆರ್ ರಿಂಕಲ್ ಸೀಡ್ಸ್ ಈ ತರ ತೆಗೆದುಕೊಂಡಿದ್ದೇವೆ. ಓಕೆ, ಸೊ ಹಿಯರ್ F1 ಪ್ಲಾಂಟ್ಸ್ ಯಾವ್ದು ಪ್ರೊಡ್ಯೂಸ್ ಆಯ್ತು? F1 ಪ್ಲಾಂಟ್ಸ್. ಸೊ ಜಸ್ಟ್ ಅ ಮಿನಿಟ್, ಗ್ಯಾಮೆಟ್ಸ್ ಯಾವ್ಯಾವ್ದು ಪ್ರೊಡ್ಯೂಸ್ ಆಗತ್ತೆ ಅಲ್ವಾ? ಫಸ್ಟ್ ದೇ ಪ್ರೊಡ್ಯೂಸ್ ದ ಗ್ಯಾಮೆಟ್ಸ್. ಇಲ್ಲಿ ಕ್ಯಾಪಿಟಲ್ ವೈ ಇರತ್ತೆ, ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಇರತ್ತೆ ಗ್ಯಾಮೆಟ್ ಅಲ್ಲಿ, ಆಲ್ ದ ಗ್ಯಾಮೆಟ್ಸ್. ಅಂಡ್ ಹಿಯರ್ ಸ್ಮಾಲ್ ವೈ ಸ್ಮಾಲ್ ಆರ್. ಸೊ ದೀಸ್ ಆರ್ ದಿ ಗ್ಯಾಮೆಟ್ಸ್. F1 ಜನರೇಷನ್ ಅಲ್ಲಿ ಏನಾಗತ್ತೆ? ಸೊ ದೀಸ್ ಗ್ಯಾಮೆಟ್ಸ್ ಫ್ಯೂಸ್. ಕ್ಯಾಪಿಟಲ್ ವೈ ಸ್ಮಾಲ್ ವೈ ಕ್ಯಾಪಿಟಲ್ ಆರ್ small r. So this is the F1 generation plant. What is the phenotype? What is the character of the F1 generation? Can you comment? F1 generation plant ಯಾವ ತರ ಇರುತ್ತೆ? ಯಾವ ತರ ಸೀಡ್ಸ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ? ರೌಂಡ್ ಯೆಲ್ಲೋ ಸೀಡ್ಸ್ ಅಥವಾ ರೌಂಡ್ ರಿಂಕಲ್ ಸೀಡ್ಸ್? ಯಾವ ತರ ಸೀಡ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ? ಕಮೆಂಟ್ ಮಾಡ್ಬೋದಾ? F1 ಪ್ಲಾಂಟ್ Can you comment? ಓಕೆ, ಇಲ್ಲಿ ಕ್ಯಾಪಿಟಲ್ ವೈ ಇದೆ ಅಲ್ವಾ? ಕ್ಯಾಪಿಟಲ್ ವೈ ಇರೋದಕ್ಕೆ ಯೆಲ್ಲೋ, ಇಲ್ಲಿ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಇದೆ, ಸೊ ದಟ್ ಈಸ್ ಡಾಮಿನೆಂಟ್. ರೌಂಡ್ ಯೆಲ್ಲೋ ರೌಂಡ್ ಕಲರ್ ಸೀಡ್ ಪ್ರೊಡ್ಯೂಸಿಂಗ್ ಪ್ಲಾಂಟ್ಸ್ ಆರ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಇನ್ ದ F1 ಜನರೇಷನ್. ಈಗ F2 ಜನರೇಷನ್ ಪ್ಲಾಂಟ್ ಅನ್ನ ಆಬ್ಟೈನ್ ಮಾಡಬೇಕು ಅಲ್ವಾ? ಸೊ ದಟ್ ಈಸ್ ಕ್ವೈಟ್ ಡಿಫಿಕಲ್ಟ್. ಸೊ ವಿ ಹ್ಯಾವ್ ಟು ಯೂಸ್ ದ ಪನೆಟ್ ಸ್ಕ್ವೇರ್ ಟು ದಿಸ್. ಇಲ್ಲಿ ನೋಡಿ ಯಾವ ತರ ಗ್ಯಾಮೆಟ್ಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗತ್ತೆ ಅಂತ ನೋಡಿ. ಹಿಯರ್ ಕ್ಯಾಪಿಟಲ್ ವೈ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಒನ್ ಗ್ಯಾಮೆಟ್, ಅಂಡ್ ಕ್ಯಾಪಿಟಲ್ ವೈ ಸ್ಮಾಲ್ ಆರ್ another gamete, and small y capital R and small y small r. So these are the gametes produced. And ಈ ಗ್ಯಾಮೆಟ್ಸ್ ಏನಾಗತ್ತೆ, ಯಾವ ತರ ಕಾಂಬಿನೇಷನ್ ಆಗತ್ತೆ ಅನ್ನೋದನ್ನ ಚಕ್ಕರ್ ಬೋರ್ಡ್ ಯೂಸ್ ಮಾಡ್ಕೊಂಡು ನೋಡೋಣ. ಸೊ ಐ ವಿಲ್ ರೈಟ್ ದ ಗ್ಯಾಮೆಟ್ಸ್ ಹಿಯರ್. ಇಲ್ಲಿ ನೋಡಿ ಗ್ಯಾಮೆಟ್ಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗಿರೋದನ್ನ F1 ಜನರೇಷನ್ ಅಲ್ಲಿ ಗ್ಯಾಮೆಟ್ಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗಿರೋದನ್ನ ಈ ಒಂದು ಕಾಲಮ್ ಅಲ್ಲಿ ಬರಿಬೇಕು ಈ ತರ. ಓಕೆ, ಕ್ಯಾಪಿಟಲ್ ವೈ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಸ್ಮಾಲ್ ವೈ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಕ್ಯಾಪಿಟಲ್ ವೈ ಸ್ಮಾಲ್ ಆರ್ ಅಂಡ್ ಸ್ಮಾಲ್ ವೈ ಸ್ಮಾಲ್ ಆರ್ ಈ ತರ ಗ್ಯಾಮೆಟ್ಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗತ್ತೆ. ಇದನ್ನ ಎರಡು ಕಾಲಮ್ ಅಲ್ಲಿ ಬರಿಬೇಕು. ನೆಕ್ಸ್ಟ್ ನೋಡಿ ಇದನ್ನ ಆಡ್ ಮಾಡಿ ಬರೀರಿ. ಫಸ್ಟ್ ಒನ್ ದಿಸ್ ಒನ್ ಕ್ಯಾಪಿಟಲ್ ವೈ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಆಯ್ತು ಅಲ್ವಾ? ನೆಕ್ಸ್ಟ್ ಇದು ಕ್ಯಾಪಿಟಲ್ ವೈ, ಹಿಯರ್ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಇಸ್ ದೇರ್, ದೆನ್ ಕ್ಯಾಪಿಟಲ್ ವೈ ಸ್ಮಾಲ್ ಆರ್. ಸೊ ನೆಕ್ಸ್ಟ್ ದಿಸ್ ಒನ್, just combine it and write. Capital Y anna first ಬರ್ಕೊಳ್ಳಿ. Capital Y, capital R, here small r is there. ಇಲ್ಲಿ capital Y ಇದೆ. Next this one. ಈ ತರ ಕಂಬೈನ್ ಮಾಡ್ತಾ ಬರಿತಾ ಹೋಗ್ಬೇಕು. So this thing you already learnt in the 10th standard ಅಲ್ವಾ? So ಈಗ ಫಸ್ಟ್ ಒಂದ್ ರೋ ಆಯ್ತು. ನೆಕ್ಸ್ಟ್ ರೋ ನೋಡಿ. ನೆಕ್ಸ್ಟ್ this gamete with the remaining 4. ಏನಾಗತ್ತೆ? ಕ್ಯಾಪಿಟಲ್ ವೈ ಸ್ಮಾಲ್ ವೈ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಅಲ್ವಾ? ದೆನ್ ನೆಕ್ಸ್ಟ್ ಒನ್ ಸ್ಮಾಲ್ ವೈ ಕ್ಯಾಪಿಟಲ್ ಆರ್. ದೆನ್ ವಿತ್ ದಿಸ್ ಒನ್ ಕ್ಯಾಪಿಟಲ್ ವೈ ಸ್ಮಾಲ್ ಆರ್. ಅಂಡ್ ದ ಲಾಸ್ಟ್ ಒನ್ ಸ್ಮಾಲ್ ವೈ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಸ್ಮಾಲ್ ಆರ್ ಅಲ್ವಾ? ಸೊ ದೆನ್ ಟೇಕ್ ದ ಥರ್ಡ್ ಗ್ಯಾಮೆಟ್. ಸೊ ಈ ಒಂದು ಗ್ಯಾಮೆಟ್ ಅನ್ನ ಇದ್ರ ಜೊತೆ ಮಲ್ಟಿಪ್ಲೈ ಮಾಡಿ ಬರಿಬೇಕು ಅಲ್ವಾ? ಸೊ ಇಲ್ಲಿ if that gamete is having the, if that zygote is having the capital Y idre, that is yellow. Capital R idre, that is round. Okay, ಇಲ್ಲಿ ನೋಡಿ, here round yellow seed producing plant, round ಮತ್ತೆ yellow seed producing plants ಎಷ್ಟು ಪ್ರೊಡಕ್ಷನ್ ಆಗಿದೆ ಟೋಟಲ್? 1 2 3 4 5 6 7 8 9 ಟೋಟಲ್ ರೌಂಡ್ ಯೆಲ್ಲೋ ಸೀಡ್ಸ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗಿರುವಂತ ನೋಡಿ, ರೌಂಡ್ ಯೆಲ್ಲೋ ಸೀಡ್ಸ್. ಸೊ ದಟ್ ಈಸ್ ಸಿಮಿಲರ್ ಟು ದ ಪೇರೆಂಟ್ ಅಲ್ವಾ? ರೌಂಡ್ ಯೆಲ್ಲೋ ಸೀಡ್ಸ್ 9 ಇದೆ. ದೆನ್ ರೌಂಡ್ ಗ್ರೀನ್ ಸೀಡ್ಸ್, ರೌಂಡ್ ಗ್ರೀನ್ ಸೀಡ್ಸ್ ಎಷ್ಟಿದೆ? ದಟ್ ಈಸ್ 3. ದೆನ್ wrinkled yellow seeds, that is 3, and wrinkle green seed ಎಷ್ಟಿದೆ? 1 ಇದೆ. ಜಸ್ಟ್ ಸಿ ದ ಪೇರೆಂಟಲ್ ಕಾಂಬಿನೇಷನ್. ಪೇರೆಂಟಲ್ ಕಾಂಬಿನೇಷನ್ ಅಲ್ಲಿ ಏನಿತ್ತು? ರೌಂಡ್ ಯೆಲ್ಲೋ ಸೀಡ್ಸ್ ಇತ್ತು ಅಂಡ್ wrinkle green seeds ಇತ್ತು ಅಲ್ವಾ? here wrinkle green seeds, wrinkle green seeds. ಬಟ್ ರೌಂಡ್ ಗ್ರೀನ್ ಸೀಡ್ಸ್ ಮತ್ತೆ ರಿಂಕಲ್ ಯೆಲ್ಲೋ ಸೀಡ್ಸ್ ಏನಾಗಿರ್ಲಿಲ್ಲ, ಪೇರೆಂಟಲ್ ಜನರೇಷನ್ ಅಲ್ಲಿ ಇರ್ಲಿಲ್ಲ ಅಲ್ವಾ? ಪೇರೆಂಟ್ಸ್ ನಾವು ಯಾವ್ದು ತೆಗೆದುಕೊಂಡಿದ್ವಿ? ರೌಂಡ್ ಯೆಲ್ಲೋ, ರಿಂಕಲ್ ಗ್ರೀನ್. ಸೊ ಇಲ್ಲಿ 2 ಇದೆ, 2 ಹೊಸ ಕಾಂಬಿನೇಷನ್ ಫಾರ್ಮೇಶನ್ ಆಗಿದೆ ಅಲ್ವಾ? ರೌಂಡ್ ಗ್ರೀನ್ ಸೀಡ್ಸ್ ಪ್ರೊಡಕ್ಷನ್ ಮಾಡುವಂತ ಪ್ಲಾಂಟ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗಿದೆ, ರಿಂಕಲ್ ಯೆಲ್ಲೋ ಸೀಡ್ ಪ್ರೊಡಕ್ಷನ್ ಮಾಡುವಂತ ಪ್ಲಾಂಟ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗಿದೆ. ಸೊ what we can say, ಇಲ್ಲಿ ನೋಡಿ, ಪೇರೆಂಟಲ್ ಜನರೇಷನ್ ಅಲ್ಲಿ 2 ಇತ್ತು, ಬಟ್ ಹಿಯರ್ we can see the 4 types of the plants. So what it explains, when 2 characters are combined in a hybrid, segregation of one pair of traits is independent of the other pair of traits. Here segregation of the trait, ಈಗ ನೋಡಿ ಯೆಲ್ಲೋ ರೌಂಡ್ ತೆಗೆದುಕೊಂಡಿದ್ವಿ ಅಲ್ವಾ? ಯೆಲ್ಲೋ ರೌಂಡ್, ಗ್ರೀನ್ ರಿಂಕಲ್ ತೆಗೆದುಕೊಂಡಿದ್ವಿ ಅಲ್ವಾ? ಇಲ್ಲಿ ಏನಾಗಿದೆ? ಯೆಲ್ಲೋ ಅಂಡ್ ರಿಂಕಲ್ ಇಸ್ ಕಂಬೈನ್ಡ್, ರೌಂಡ್ ಅಂಡ್ ಗ್ರೀನ್ ಇಸ್ ಕಂಬೈನ್ಡ್ ಅಲ್ವಾ? ಸೊ ದಿಸ್ ಈಚ್ ಕ್ಯಾರೆಕ್ಟರ್, ರೌಂಡ್ ಅನ್ನುವಂತ ಕ್ಯಾರೆಕ್ಟರ್ ಗ್ರೀನ್ ಜೊತೆ ಕಂಬೈನ್ ಆಗಿದೆ, ಯೆಲ್ಲೋ ಅನ್ನುವಂತ ಕ್ಯಾರೆಕ್ಟರ್ ಏನಾಗಿದೆ? ರಿಂಕಲ್ ಸೀಡ್ಸ್ ಜೊತೆ ಕಂಬೈನ್ ಆಗಿದೆ. ಸೊ ದಿಸ್ ಈಚ್ ಕ್ಯಾರೆಕ್ಟರ್, ಸೊ ದಟ್ ಈಸ್ ಇಂಡಿಪೆಂಡೆಂಟ್ ಆಫ್ ಅದರ್ ಪೇರ್ ಆಫ್ ಕ್ಯಾರೆಕ್ಟರ್. ಇಲ್ಲಿ ಯೆಲ್ಲೋ ಮತ್ತೆ ರೌಂಡ್ ಅನ್ನುವಂಥದ್ದು, ದೀಸ್ ಆರ್ ಇಂಡಿಪೆಂಡೆಂಟ್, ದೇ ಆರ್ ನಾಟ್ ಕಂಬೈನ್ಡ್, ದೇ ಆರ್ ನಾಟ್ ಜಾಯಿಂಟ್, ದೇ ಆರ್ independent of each other. So that is the law of independent assortment. Okay, so inheritance of the 2 gene. So this is very important for your exam. Okay, so if you have any doubt, you can write down in the comment box. I hope nimge artha agide anta ankoltene. Thank you for watching. So please don't forget to comment how was the class. Please comment it. If you have any doubts or any problems, you can write down in the comment box. Thank you. We'll continue in the next class. Okay.